Last lesson, we talked about karyotyping. We said that a karyotype is a picture of the human genome. We said that, what else did we say? We said that chromosomal abnormalities can exist. That's when you have either too many chromosomes or you have too few chromosomes. And then what we did is we said that at the end, we said a mutation is, yep, one from mom, one from dad. Yes, they're correct, David. And we said that a mutation is when one of these DNA goes bad. It does the wrong thing, and the wrong thing gets made and said and sent. So that wrong thing then can mean that you will get the wrong DNA. You might get, and the wrong DNA can be caused by these kinds of things, and that wrong DNA can mean you might get an abnormal protein or no protein at all. Today's lesson, we're going to be talking about mitosis. And we don't have much time left in today's lesson, so we may not finish all of it, but I think it's worth trying to get through some of it. And then I'm going to give you guys some homework that I'm going to need you guys to wrap up. So I'm going to give you guys to this. So can I get you guys to jot down lesson 41? Gail says she couldn't find lesson 40 last lesson because, and I think that's probably because my computer hasn't been updating properly to the server, but hopefully I will have a look at that this afternoon. I'll see if I can get it fixed. Okay, so mitosis. We will be able to talk about, describe the process and stages of mitosis. Now, if you remember, we've already discussed mitosis. We said that mitosis is when a cell splits into two identical cells. Now, what we're going to be doing is we're going to be talking about how this process works. We're going to go into a little bit more detail than we did when we talked about last time. So let's start off with just a quick refresher about what mitosis is. Why do we make new cells? That's one of the really big questions that I thought I would start off with. Why do we need new cells? Why do we need mitosis in the first place? So mitosis is mainly used for two main things: repair and growth. Let's start with repair. Over time, as cells die or get damaged, and this can be caused by cuts or sunburns, but also when their cells get overused. If you have if you've got a cell in your, if you've got a muscle cell, and that muscle cell its job is to open, close, and contract. And then let's just say that you do a lot of running and that muscle cell gets worn out. Essentially, that muscle cell is just going to die. And you need to replace that muscle cell. You need to repair the body. If you've been running or working out and you feel you get like afterwards your body is sore, That's because you've essentially destroyed a bunch of cells. But you might think, oh my God, but it's okay. You'll actually get new cells grow. And if you've been working out hard enough, those new cells that show up will be stronger and hardier than the cells that used to be there. And that's, you might even get more cells there. That's how you get bigger, stronger muscles. And of course, if you cut your arm or your leg when you're running, your body will grow new cells to replace the skin cells so that they can do it. The other type of reason why is for growth. To make a person taller, you need to make more cells in your bones, your skin, your muscles, so the new cells are needed. So if you look at a baby when it's being formed, starts off with one cell, but then the cell splits and makes 2 copies, makes 4 copies, makes 8 copies, 32, 64, 128, so on and so forth cells. And all of these cells make it grow. You can add more cells in your bones, which will make you taller, and so on and so forth. So mitosis is how the body makes new cells. It's how these mitosis allows us to grow and to keep going, to repair our body as we get older. If you had cells that were able to undergo mitosis a lot faster, maybe you could be heal from wounds a lot faster. And this is why when we talked about mutations, I drew a picture of Wolverine, because it's that's what the whole process is that it's the whole process of new cells. I'm going to change the slide in about, I don't know, say 30 seconds. So if you haven't got all of this down, that's okay. Just get the main points. Now, I actually think this is probably the most important slide. It's a good thing that it comes first, because ultimately understanding why things undergo mitosis is really important. I explained to you guys about what mitosis is, but it's that's not useful unless you know why we actually do it. So what is mitosis again? Well, let's go through a quick refresher. Mitosis is the act of splitting a cell into 2 new cells that are exact copies or clones of each other. Before the copy can happen, the DNA is first bunched up into packets called chromosomes. These chromosomes are copied. Then the cell splits in a process called cytokinesis. When this happens, each new cell gets 1 copy of the chromosomes. Now, you don't need to copy down the picture. You don't need to know the names of the different sections and stuff. You just need to know at least that, you get 2, the main parts of this slide is that you get 2 exact copies. You don't even need this bit here. You can just write 2 exact copies. So for those of you guys that are listening to me talk while you guys are putting it down, on the right, so chromosomes, remember, normally the DNA is in nice, really long strings, but when it's when the cell needs to undergo mitosis, these really long strings all sort of bunch up together. when those really long strings bunch up together, they get called chromosomes. And the first step is that you double the chromosomes. So you can see here, the chromosomes get
So is this
doubled.
on Google?
Should
Google.
be.
All right.
After the chromosomes get doubled, you then get a process where the doubles get split. And that's the main thing that I want to show you guys today. After I've finished with this slide, it's just this idea here that you double the chromosomes and then you split them. So you like double them, so you go from having 46 strings to 92 strings, and then you split them. One cell gets 46, the other cell gets 46. And then those cells go off and they do their own thing. They split again, and so on and so forth. Let's change the slide in about 30 seconds. And by the way, as I said, I love this word, cytokinesis. Do you need to know this word for the test? No. Is it a cool word to know? Yes. I think it's a cool scientific word to know. It's a cool scientific word to say. I love when I find words that just, chirality, cytokinesis. It's just like, ooh, it sounds fancy. It sounds like some kind of, villain from like the MCU or something like that. I don't know. This is me thinking out loud. This is what happens when you let me talk to myself. So let's grab a look at the process, and then we'll do the last little bit of copying down. So the process looks like, actually, no, there is a big thing that I need you guys to do. How does DNA copy? So we haven't discussed this. So this is wrong. Let me just cross this out. How does DNA copy? Well, the first thing is you take a DNA strand, right? And the first thing it does is it unzips. So I want you guys to watch this. So don't worry about copying it down for the moment. Just watch this. First thing we do is we unzip the DNA, right? You might say, why am I using the word unzip? Because in my opinion, it is like it's like a zipper. You are unzipping the DNA. And then what happens is that we add a bunch of DNA in there. We know that T matches with A. So we can replace that T with an A. We can replace that C with an A. And you can look here, right? TCA. TCA. It matches. So, and I've just drawn this left side in red because it just makes it easy. And what happens after this is you'll end up with 2 new strands of DNA. So let's go through again. How does this work? Step 1, you unzip the DNA. Step 2, you replace some of these bases. And step 3, you end up constructing 2 strands of DNA. And the reason why we're able to construct these strands of DNA so well is because we know that T always bonds with A. So if I split these up, then T will bond with A again. And so you can actually copy these really easily. So what are the 3 steps? Someone said after you finish teaching. What's he saying? Do we copy it? Yes, now I want you to copy it. So thank you guys. You guys are working well together. The 3 steps are: step 1, the DNA unzips. Step 2, Matching base pairs move in to match the bases. In step 3, 2 new DNA strands. If you've already copied that down, what you can do is you can maybe draw a little picture. You do not have to, but I'm going to draw a little picture underneath. You do not need to draw this. I'm just going to draw this because 1, I'm bored, and 2, because some of you guys may have finished copying it already. Okay, there you go. You do not need to draw that. If you finish writing it down though and you're bored, you can. I'm going to change the slide in about 10 seconds. So, or maybe more, maybe 30 seconds, but yeah. Okay. Now, how does this work? So there's nothing to copy down here. I just want you to watch. Here are 46 chromosomes just before copying. They have 1 strand of DNA, so they are called a chromatid. Each chromosome is part of a group of 2 or a diploid. Again, it's weird. So each of these is called a chromatid. If you count them, there are 46. If you don't want to count them, believe me. So each of these comes in a group of 2. Okay. Now, the first thing that happens is that each of these strings of DNA makes an exact copy. And that will look like this. Wait for it. Bang. After copying, each of the 46 chromosomes is made of 2 chromatids. Now, this is really weird, okay? Because the reason that this is weird is this. You guys can see that all of these are double, but there are still 46 chromosomes. And the reason for that is if you count each of these, there are still 46 of them. But each of these is made up of 2 chromatids. So therefore there are 92 chromatids. There is double the amount of chromatids. There is double the amount of strings. So we've gone from, let's just go back a sec. We've gone from 46 chromatids and 46 chromosomes to 46 chromatids. No, 46 chromosomes, but 92 chromatids. We've doubled it. Now this is going to get a bit confusing because they're all blue. So let's make this a little bit easier for us to deal with. Let's make some of these red. Now if I make some of these red, it's still 40, there are still 92 chromatids, right? And there are still 46 chromosomes. But now that we're in red, I can tell them apart. And now that they're in red, I can actually now split these cells. I'm going to split the cells. We're going to do some cytokinesis, and we're going to split the cells like this. Split the DNA like this. each DNA, if you watch, each DNA gets one copy of the first, one copy of the second, one copy of the third, one copy of the fourth, etc., etc. And so what you end up with is 2 new cells. And each cell has one copy of each chromosome because they are exact copies. Let's go through this one more time. So we start with all 46 chromatids. We then double them to get to 92 chromatids. And then we split the 92 chromatids with half and half, each getting one copy of each chromatid, each chromosome. One copy of chromosome 1, one copy of chromosome 2. And that makes 2 new cells. Now, we're almost out of time. I'm not going to get a chance for you guys to copy this down, I don't think. But what I want you guys to copy down instead is I'd like you to write this down. Ladies and gents, this is the homework I'm going to set. Now, this is from the Pearson chapter 2.1. Now, before I get half of you guys saying, but sir, I left my textbook at school, I've also got a link here to the actual chapter that you guys are going to be doing. So you guys are going to be doing questions 1, 2, 4, 5, 7, 9, 10, 13. They're going to be due Friday. So you've got a fair bit of time to get them done.
Sir.
Gail, don't worry. We'll copy. Someone just said, can you go back a slide? Don't worry. We're going to copy down that last slide probably, which is whoopsies, where is it? Whoopsies. We're gonna copy down this.
Are we going to be working on those questions at all in class?
I might be able to give you a little bit of time on Thursday to work on it, but you should be focusing on it. You should be doing a little bit of it on tonight and tomorrow. So I would say if I was you, I'd try to get up to question 7 before Thursday, just in case I don't give you any time. Most of these are multiple choice and pretty easy questions. Sorry, Zane, go.
We upload it onto your thingy. Sorry, Google Classroom.
I will put on the Google Classroom, but this link will be also on the Google Classroom too, so you can actually click on it. So I'll do that too. Yeah, and this slide here as I said, this slide here that I skipped over, we'll copy this slide down tomorrow just because I don't have enough time. I wanted you guys to see this so that you didn't go, oh my God, sir. I wanted to make this clear. So that'll be due Friday. So I at least want you to get up to question 7 before Thursday, because then that way, if I don't give you time in class on Thursday, you've got, you almost finished anyway, but you should be working on it outside of class as well. That's it for me, because it's now 10:36. It's recess for you guys. I will see you guys on Thursday. We will work on meiosis, which is the opposite of mitosis.
Bye.
On Thursday.
Bye, sir.
See you later.
Sure.